This is most students' least favorite lecture in the semester. I still think it's probably worse to do genes and DNA and RNA, but most students, because the brain is so complex, because the brain is so difficult, a lot of students hate this lecture. This is going to be broken up into 4 different parts. But first, we're just going to do the brain introduction and some circulation. How does blood and fluid move around the brain? Part 2 will be parts and structures and functions. So part 2 is what you're going to be really the meat of this lecture and what you need to memorize for the test, because I'm interested in different parts of the brain and what they do. Part 3 is going to be different types of functions of the brain, that how they connect and what those connections are doing. And then finally, part 4 is the shortest one, but also very important because it's going to look at the 12 cranial nerves. So if we just take a basic look at this picture, and you'll see this picture on each of the next 4 micro lectures, but if you kind of look at this, all this tissue kind of looks the same, but over here it looks different. And the reason why this looks different is because this is a growth of abnormal tissue and cells, and that's just to look at brain tumor. And you can see how there's different types of coloration in there. Those can be blood vessels and blood supply to the brain. This right here can be how the cells are actually growing. But without knowing what type of tissue this is, even is this necrotic tissue, is it dead, or is this patient, did this patient die of this, or what? It's kind of impossible to know, but you can clearly see this tissue doesn't look like the rest of it, so this tissue is cancerous. So introduction, the brain is complex. I don't need you to, you don't need me to tell you that. The brain function is associated with life. Breathing, moving, living, that's all happening in the brain. But more importantly, imagination, memories, all of these things also are going to be involved in the brain and are probably the most important part of life. In this chapter, we're going to study the brain and finally the cranial nerves, and then see how they are all connected. We're going to provide some insight into brain circuitry and function in the second part, but for the most part, I want to introduce you to the brain. So first, Aristotle thought the brain was a radiator, and all that meant was it just cooled blood, just like a radiator would in your car. He was wrong. Hippocrates was more accurate. From the brain only arises our pleasures, joys, laughter, jest, as well as our sorrows, pains, griefs, and tears. So he had a better idea of that. If the brain activity stops, you're dead. And that's the clinical criteria for death, is no brain activity. So once the brain activity stops, even if you're able to be on a respirator and a life support system, if you don't have brain activity, you're not clinically alive. An evolution of central nervous system allows spinal cord has changed very little when the brain has changed a great deal. So how we send signals hasn't changed. The brain, on the other hand, how it deals with all these inputs has changed a lot. And the greatest growth, vision. Our vision is completely different than other animals. The way we're able to memorize things and have memories is different than other animals. And then motor control and our ability to have a thumb, that has changed a lot. So in this lecture, major subdivisions and anatomical landmarks of the brain. We're going to locations of the gray and white matter. Gray cell bodies, white matter is going to be axons with myelin. So those are going to be processing areas, conductive areas. We're going to quickly go over embryonic development of the central nervous system and how this relates to adult brain anatomy. Most students in this lecture are going to have a difficulty with the embryonic development. And you might not understand why embryonic development is important for the brain, but understanding how different parts of the brain come from the different embryonic cells can lead to better understanding of how those systems work, how those systems talk to other systems, and if you have a disease or you have some type of genetic problem with one area, you're likely to have a genetic problem with another area if they all come from the same embryonic cells. So let's get started. Front, back. Rostrals towards the forehead, caudal toward the spinal cord. Caudal means tail, so spinal cord looks like a tail, rostral to the nose or forehead. You have three major portions: cerebrum, cerebellum, brainstem. You have the cerebrum, which contains the cortex, cerebellum, which is this little thing that looks like a bunch of little fibers kind of wound together, almost like a bird's nest, and the brainstem in the bottom. The brainstem comes up here right in front of the cerebellum also. You have a longitudinal fissure. Longitudinal fissure separates the left and the right hemispheres. You have a gyri. The gyri are the thick folds. You can see there's one up here, there's one back there, and in between those two that I showed you, there's a sulcus between them, and the sulcus is that shallow groove. The one that comes right before this central sulcus is important, and the one that comes after that central sulcus is also important. So think about that one line that runs down the middle like that, and when we get to it again, you'll remember a gyri is a fold, sulci is the groove. Corpus callosum you can't even see. Corpus callosum is in between, and we'll see it when we look at the sagittal section, and the thick nerve bundle at the bottom of the longitudinal fissure that connects the hemispheres. So this will kind of look almost like an ear. And when we look at the sagittal section, I'll show you again the corpus callosum. Brainstem we mentioned, it's kind of hidden there, but you can see the brainstem is all that in yellow. And it's pretty much what would remain if we remove the cerebrum and the cerebellum. The major components of the brainstem: the diencephalon, the midbrain, pons, and the medulla oblongata. The diencephalon, midbrain, pons, medulla oblongata, we have studied these a lot, so we know a lot about what each of those components do. So we're going to spend some time looking at those major structures. And here's the sagittal section. You'll learn most of this stuff in lab, so I'm not going to go over it now, but here's the corpus callosum that I showed you. There's your cerebrum up there. You can see the central sulcus right there between the parietal, or the frontal and the parietal lobes. You can see your cerebellum back here. You can see those white lines. Those white lines are going to be the arbor vitae. You can see the midbrain, the pons, and the medulla oblongata right down there. This area right here is actually just a space, because this is where cerebral spinal fluid is going to be going with the ventricles. We'll look at later. And right below this is a commissure that connects the right and the left brain. Below that is your hypothalamus. Behind it is your thalamus. And because this is a space, you have a thalamus on the left, a thalamus on the right, a hypothalamus on the left, a hypothalamus on the right. Below the hypothalamus is your optic chiasm. This is where your optic nerve crosses or decussates, or the left goes to the right. And below that is your pituitary gland, which is going to be very important when we talk about the endocrine and exocrine system and circulation in the body in AMP2. So we went through most of those. Let's start looking at what's gray, what's white. So gray versus white. So gray, cell bodies, dendrites, synapses. They're gray because they don't have a lot of myelin. And because they don't have a lot of myelin, these are going to be, nuclei deep within the brain. Because they have the cell bodies, this is going to have the different processing of the brain. If it's white, or there's tracks, they're white or in their tracks because there's axons. And because they have axons, they're carrying information somewhere, and they use myelin to do that, which gives them a white color. So, embryonic development. Nervous system develops from the ectoderm. The ectoderm is the outermost tissue layer of the embryo. Early in the third week of development, the neuroectoderm, neuron, ectoderm, outermost layer, dorsal streak appears along the length of the embryo, all the way in the third week. It thickens to form the neural plate. This is destined to give rise to most neurons and all glial cells except for microglia, and they're microglia because these are macrophages from the blood. So as thickening progresses, the neural plate sinks and its edges thicken. So it starts to form like a groove. That forming a neural groove with raised neural fold on each side. The neural folds will then fuse along the midline. This is the beginning of the cervical region and progressing rostrally and caudally from the nose to the tail. By the 4th week, you now have a hollow channel. This channel is called the neural tube. The neural tube separates from overlying ectoderm. It's going to go deeper. It grows lateral processes that will later form motor nerve fibers. The lumen of the tube becomes fluid-filled space. Central canal in the spinal cord, ventricles of the brain, that's going to be containing that fluid. Notice none of this is in red. Now we're in red. So, okay, neural crest forms, ectodermal cells that lay along the margins of the groove separate from the rest, forming a longitudinal canal on each side. So now we're going to start seeing different meninges. We're going to start to see different cells. And by the 4th week, the neural tube is going to show 3 anterior dilations or primary vesicles. So 3 different types of bulges. The forebrain, the midbrain, and the highbrain. This is important because the forebrain, the prosencephalon, starts to form other things. Midbrain, or the mesencephalon, starts to form pretty much just the mesencephalon. Talk about that later. In the hindbrain, the rhombencephalon starts to differentiate into other structures. Forebrain, the telencephalon, and the diencephalon. If you remember, the diencephalon is part of the brainstem. The telencephalon becomes the cerebral hemispheres. The telencephalon is very important because without the cerebral hemispheres, you don't have the left and the right cerebral hemispheres. You don't have a lot of processing of anything. The diencephalon, optic vesicles that become the retina of the eye. That's very important because you won't have vision. Midbrain becomes undivided, just becomes the mesencephalon. In the highbrain, metin and myelencephalon. And here you can see 1920 in 2 weeks, where you can start seeing it folds, starts going up. It has thickened ends that I talked about until they start to form together, until it starts to form a neural tube. So here we go. 4th week, forebrain, midbrain, hindbrain. 5th week, we have these 5 structures. Telen, dien, mesin, metin, and myelencephalons. When you look at it, telencephalon, what does it become? Cerebral hemispheres. Diencephalon becomes the diencephalon, and it starts to form the optic nerve in the retina. The mesencephalon just stays there, it becomes the mesencephalon. And the metin and myelencephalon become the medulla oblongata. And the metencephalon becomes the pons and the cerebellum. So those are very, very, very important. And if you wonder what those 3 bulges are, you can kind of see them right there. And this is what it looks like when fully developed. Now, we're not going to go into any more detail other than that because you can get a PhD on just one of those embryonic developments. You can study your entire life how one thing forms into something else. But knowing how the full brain comes from those structures in the 4th to the 5th week until adult is very important for learning how each of these systems works together. Because the connections in the cells are going to be very similar between the different structures that come from the embryonic development. So finishing up, meninges, ventricles, cerebral spinal fluid, and blood supply. Meninges of the brain, dura mater, arachnoid matter, pia mater we looked at. We're going to review it. We're going to look at the fluid-filled chambers of the brain, production, circulation, and function of that fluid in those chambers. And then we're going to explain the significance of the blood-brain barrier. So the meninges again, from superficial to deep, dura mater, arachnoid matter, and pia mater. Dura mater is called the tough mother. If you have the chance in the in-person lab to see this, try to cut it with scissors or scalpel, or try to tear it with your hands. It's very tough. That's why it's called tough mother in German. And the pia mater is very see-through and just kind of protects the brain and covers the brain and spinal cord. The arachnoid matter is transparent. It's attached to the dura mater. They have the subarachnoid space that has the cerebral spinal fluid, which is the main reason why I'm reviewing it right now. And the pia mater again is translucent and see-through, and it's pretty much just seen on top of the brain and the spinal cord. And here's just another look of it, where you can see the dura mater, you can see the arachnoid matter here, you can see those little fibers holding it onto the pia mater right there. And that space between it is where your cerebral spinal fluid, between the arachnoid and pia mater, that's where the cerebral spinal fluid is going to be flowing around. So meningitis is going to be the inflammation of these meninges. This is very, very serious. Between 3 months and 2 years of age, if not caught, the person could die within hours. And this is caused by a bacteria or a virus that's actually able to get into the central nervous system by the nose or the throat, because during that time the blood-brain barrier is kind of there, but there are pathways with the mucous membranes, between the mucous membranes in the nose and the throat, for these viruses to get into the central nervous system. So what happens, bacterial meningitis causes swelling of the brain, enlargements of the ventricles, and possible hemorrhaging. And what happens with meningitis is it causes stiffness. Because you increase the pressure of the brain, what happens with it is stiff neck. You can't really move your neck because your neck starts to hurt. You don't understand why. Because the neurons there can't send signals very good, because the cerebral spinal fluid is pushing on the cranial nerves, pushing on the spinal cord, and pushing the brain. It'll eventually cause coma and death because of the pressure in the brain keeps building up and up and up. And the only way to diagnose this is through a lumbar puncture or a spinal tap that actually is able to draw some subarachnoid, some cerebral spinal fluid, and then you can test, is there an infection in there? And this is the ventricles that it kind of gets into, where it kind of look like ram horns, right? You have lateral ventricles, then you have one in the middle. You have a little pathway between the one down here. And if you're kind of looking at the words and looking at the names, you see these are lateral ones, but there's 2 of them. So let's just call this 1 and 2. The space right here that we saw when we look at the sagittal section is the 3rd ventricle. The aqueduct, or the river between the 3rd and the 4th, is the cerebral aqueduct. And then you have a 4th space right in front of the cerebellum called the 4th ventricle, and then eventually a line that runs down the back of the spinal cord called the central canal. And that is your ventricles and the cerebral spinal fluid. And it eventually kind of flows back up and goes into the ventricles, or goes into the sinus on top and back into the veins. Let's look at this. So 4 internal chambers in the brain. 2 are the lateral ventricles. They're the ram's horns. The 3rd ventricle is the narrow one that separates right below the corpus callosum, and also separates the hypothalamus and the thalamus from the left and right sides. The cerebral aqueduct is then going to connect the 3rd ventricle to the 4th ventricle, and the 4th ventricle is then going to connect to the central canal. Choroid plexus are important because these are special spongy massive blood capillaries on the floor of each ventricle. What they do is they're just there, and I'll show you a picture later on of what they're actually doing, but just right now, spongy mass of blood capillaries on the floor of each ventricles. Epidymal neuroglia, these are glial cells that line the ventricles and cover the choroid plexus, and this is where cerebral spinal fluid is going to be made. And if you kind of think, choroid plexus, spongy mass of blood capillaries on the floor of each ventricle. Okay, because they're going to be a mass of blood capillaries, you need these glial cells that sit on top of these capillaries and filter it. And by filtering the blood capillaries, that's what's going to be producing the cerebral spinal fluid. The choroid plexus provide the blood. The epidymal, these helper cells, are going to create little holes in those blood capillaries, so fluid like glucose, water, oxygen can be more important in the cerebral spinal fluid than just blood. So the cerebral spinal fluid that's made by the choroid plexus with those glial cells, it's clear, colorless liquid that fills ventricles and canals of the central nervous system. And it bathes the external surface, so all the outside and in between in the brain. The brain produces and absorbs about 500 milliliters a day. So you have a lot at any given time. 40% subarachnoid, external to the brain. 30% the epidymal lining the brain ventricles, and then 30 by the choroid plexus. So you do create in different areas. And we'll kind of look at some of those. But production begins with the filtration of blood plasma through the capillaries of the brain. These epidymal cells modify the filtrate. So the cerebral spinal fluid has more sodium and chloride, salt, than normal plasma does, has less potassium, calcium, glucose, and very little protein. So these cells that line these areas are able to filter the blood plasma to just create something that it really, really, really wants. And what happens, small amounts of cerebral spinal fluids fill the central canal of the spinal cord. They escape through 3 pores. And cerebral spinal fluid is then reabsorbed by arachnoid villi. And these are cauliflower-shaped extensions of arachnoid area, of arachnoid meninges. Goes through the dura mater and then up into the cerebral or superior sagittal sinus, which is the vein. And then the cerebral spinal fluid penetrates the walls of the villi and mixes with blood in the sinus. So this is where it's reabsorbed. And pretty much inside the brain, it's going to be made by the choroid plexus. What does it do? It provides buoyancy. And that might not sound like much, but by giving, you've all been in a bath, you've all been in the pool, you're a lot more buoyant. So the weight of the brain, because it's so big, if it just kind of sat there, it would kind of squish down. But because the cerebral spinal fluid allows some buoyancy for it, it allows it to kind of float in that liquid. It also provides protection. Protects the brain from striking the cranium when the head is kind of moved from side to side. Shaken child syndrome and concussions do occur from severe jolting or movement, but it also chemical stability. Because it has more sodium and chloride, for the sodium potassium, for the sodium and chloride to kind of move in and out of cells, and that's what the neuron's going to be moving or needing to provide excitability. It allows the neurons a way to get the nutrients it needs and also the waste away from it. And here you can kind of see everything again. I kind of say it this way, even though it can be outside here is kind of be made. You can see some over here, but here you can see all these different areas. Those are those little areas of areas that have those capillaries, but choroid plexus in each lateral ventricle on left and right. Then it's going to go into the third ventricle. In the third ventricles, there's more choroid plexus. It's going to add more cerebral spinal fluid. It goes down the cerebral aqueduct into the fourth ventricle. And then down here you can see there's more choroid plexus in the fourth ventricle, so it adds more cerebral spinal fluid. It can go down the central canal and go back up, and then it circulates the outside and around the brain. And then here you can see these little, they call them cauliflower-like projections. And what they are is they're just other types of cells that allow the cerebral spinal fluid to then go into this sinus in this area, where it can then circulate with venous return and then be returned back to the heart to be reoxygenated and have more nutrients added to it. So the blood supply and blood-brain barrier. The brain is only 2% of the whole body weight, but it receives 15% of the blood. The neurons have a high demand for ATP and therefore oxygen and glucose. So a constant supply of blood is critical to the nervous system. 10 seconds of interruption of blood flow, you would lose consciousness. 1 to 2 minutes of interruption can cause significant impairment. Going 4 minutes without blood can cause irreversible brain damage. So the blood supply, when we look at circulation in AMP2, it's very important that most of the blood, 15% of it, goes directly to the brain. And blood is also a source of antibodies, macrophages, toxins, and other harmful agents. So the brain has a special barrier. And this regulates what substances can get from the bloodstream into the tissue fluid of the brain. The 2 points that must be guarded: blood capillaries throughout the brain tissue, and the capillaries of the choroid plexus. I already told you what was covering the capillaries of the choroid plexus. Let's take a closer look at the other capillaries. So we need to protect these blood capillaries throughout the brain. There are a bunch of tight junctions between the endothelial cells that form the capillary walls. You're also, on top of those tight junctions, you're going to have astrocytes. We're going to talk about astrocytes in later in the neuron chapter when we talked about glial cells. Astrocytes have these little feet, these little perivascular feet that kind of reach out, hold on to the capillaries, and provide little spaces for stuff to go through. And astrocytes and these tight junctions are the main things that are going to produce the blood-brain barrier. They induce the endothelial cells to form tight junctions that completely seal off gaps between them. Anything leaving the blood must pass through the cells and not between them. That's important. Endothelial cells can exclude harmful substances from passing to the brain tissue while allowing necessary ones to pass. That's why certain drugs, if you take them, cannot go to the brain because they're too big, their structure is different. So you have special chemicals and special pills that you can take that actually can cross the blood-brain barrier. And finally, the blood-brain barrier and the cerebral spinal fluid barrier. This barrier protects the brain at the choroid plexus. We mentioned this. This is tight junctions between the epidermal cells, and these are those glial cells that are found at the end of these capillaries. And these tight junctions are absent from epidermal cells elsewhere, but they're only going to be found in these glial cells at these choroid plexus. And this allows a special exchange between the blood tissues and the cerebral spinal fluid. The blood barrier system is highly permeable to water, glucose, lipid-soluble substance such as oxygen, carbon dioxide, alcohol, caffeine, nicotine, and anesthetics. So if you think glucose, oxygen. Glucose and oxygen, we need that. So that's why it's higher concentration in your passing through the blood-brain barrier. And then slightly permeable to sodium, potassium, chloride, and other waste products, urea and creatine. Also, if you're wondering why you feel awake when you drink caffeine, because caffeine can pass through the blood-brain barrier and go to the brain. Smoking a cigarette with nicotine, alcohol, all of those can pass the blood-brain barrier. And that concludes the first micro lecture for chapter 14.